research, summer research competition. I don't know if you guys heard of it, but there's like this competition you can apply for a position to do research over the summer. It's a paid position. I can't remember how many thousands you get paid there, but it's a paid research position, and it's so there's a competition about it. I'll bring the specifics next class, but just so it's on your radar. You might want to check your emails. It's called I think it's the York University USRA, or ENURA, ENURA competition for summer research positions. The reason why I'm bringing it to your attention is my research group will be looking for 1 to 2 students through this program for this summer. So you got good grades. I think the competition is based on grades. Like I don't get to choose right off the bat. It's kind of based on grades. But from what I understand, the professor and the students kind of go in to apply together. So I'm kind of putting it out there that if you're interested and you might want to do a summer research experience, that competition's opening up. If you want to do it in my lab, we have funding for 2 projects. One of them is on the gene regulatory networks, so the transcription factor networks. We're trying to understand that regulate THC biosynthesis and CBD, so cannabinoid biosynthesis in cannabis. It's actually a federally funded project from the United States, but USRA students can get a position on that. The other project is about gene regulatory networks, the transcription factor networks that regulate the biosynthesis of something called glycolins, and those are anti-cancer metabolites in plants. So I'll bring more details. I'll try to remember for next Thursday or next Tuesday's class to put it up, but you should see something in your email. At least the PI, so the principal investigators, we have seen it in our email, so that's why I'm notifying you. So let me know if you're interested. Again, it's competitive. It's kind of like strictly based on grades, which is unfortunate because I think that a lot of really promising researchers don't necessarily have the best grades. I didn't have the best grades, and I would like to think that I'm somewhat decent researcher. But anyway, so it's based on grades. So it's kind of like you have to have pretty much straight A's, straight A pluses to be able to compete in this program. Again, it's not I didn't make those rules. It's just that's what the program's about. So just check your email if you're interested. USRA, you might have been emailed something from York about that. Okay, all of that non-interesting stuff aside, let's get into the genetics. So we're going to do some iClicker activities today. So get your iClicker open, logged in. I'm doing it again. I think I'm telling you to do that, and you can't get in until this will let me in. So just hold on a sec here. I'll get this up. Does anybody have any questions on the USRA? No? Yeah. Oh, you haven't seen it? Okay. Yeah, that's, so I'm not quite sure exactly. Do you remember seeing it last year and previous years? Okay, so you guys will get an announcement. It just hasn't come around yet. It's coming. It's coming soon. Okay. Okay, so the clicker, I'm logged in here. We'll get to that in a minute. I guess what I'm gonna do, I'll just start off by giving you, we'll have a little bit of extra time today, I think. I'll give you a few minutes, just about 5 minutes or so, to finish up your feedback stage of critic. Is that open to you now? Can you see that, your feedback stage? You don't need to do much. You don't need to write much. So give some feedback. So what have you guys done so far? You created your entry, right? You created tell a personal experience, something that the creation stage was to create that, and then you actually evaluated your peers, right? Does everybody have like 2, 3 evaluations? All right. Now what you're going to do is you're going to provide feedback on those valuations. So just 5 minutes. I'll give you the time now. Go. Yeah, I think it's like you write your response to your evaluations. So if you're somebody, they should say supportive things. And they should say things that help you improve your answer, like maybe your communication was unclear and this is, so you can provide some feedback on that. Yeah, right, yeah. So, try not to don't be negative and like, this sucks, your write-up is terrible. No, like, that doesn't help anybody. You need to explain why and be supportive. Oh yeah, I'm supposed to mention this too. Okay. Yeah. Okay. Guys, there's like a button, right? Like if what is it there's a button that if like you don't like the feedback you got, you can dispute it or something like that. Do not use that button. Okay, well, just let me explain. Only use that button if you have a serious complaint that you want to draw my attention to. And actually, I would prefer you come talk to me or something along those lines rather than use the button. Don't expect anything's going to be done from using that button because the only thing that a lot of people are going to be unhappy. They didn't like this comment or this or that. Yeah, okay. But if you find something harassing or threatening or really inappropriate, that's when I want you to let me know. Okay? All right.
Jeez.
Okay, just another minute or two. If you don't finish it now, you can just finish it after class. Try to take your feedback constructively, folks. I know it's hard. You need to, okay, like in the future, you're gonna have to like get used to this back and forth, iterative, you get feedbacks and it's generally to help you improve. Like, Jim, that you advised that document and to distribute to the entire company, but this needs to be changed. It's a very common thing. Oh, okay, Sally, yeah, okay, I revised this, and this. Thank you for your feedback. That kind of thing. It's just, it's a common thing in any job to get feedback, take it constructively, and try to improve. Okay, let's move on. You guys can finish that after class if we have time or outside of class. Okay, so last week, what did we do last week? We did this activity. I love looking at these things. You get so many. We're talking about cardiovascular disease. That was the exercise, CVD, cardiovascular disease. There's all kinds of. It's unclear all the details about what causes cardiovascular disease, right? Even nowadays, we know so much, and it's so intensively studied. Decades, many research groups, thousands of people, and we still don't really clearly know what causes it. And so wanted you to do this activity to, some of you might recognize, I got yours up here, but I wanted you to take all these factors, diet, genes, ethnicity, and so on and so forth, and let me know what you think as definitely impacts your likelihood of getting cardiovascular disease. And also, that'd be the solid line, so you know for sure one factor impacts another factor, or one factor impacts the cardiovascular disease directly. And the dotted lines were, I'm not quite sure, but I think there's an impact. Yeah, that's interesting. So if you look at this, I think no two groups are the same. I got a whole bunch of these here. This one's awesome. These guys really thought about it hard. Love that. Yeah. Yeah, so, it's funny because a lot of the arrows are going to cardiovascular disease, but not like if you have cardiovascular disease, does that cause you to have, I don't see a lot of arrows going in the other direction. So now if I were to tell you that, you'd probably do it over again and be all kinds of arrows going the other way too, right? So what do you think the purpose of this is? What do you think we're doing here? Why are we looking at this? Why are we trying to, what's the point of this? Yeah. Very good. Excellent answer. The point is to show that, we have all these different factors. Many factors can affect a particular phenotype. Our phenotype in this case is cardiovascular disease. And so you can imagine with all of these factors affecting this one phenotype, how difficult it can be to figure out what's causing cardiovascular disease. Look at all these factors, and they're all affecting one another, and they like that. So it's very complicated. And right, so you can imagine trying to understand what genes we got. We got tens of thousands of genes, hundreds of thousands of splice forms and variants and so on, and different alleles and so on. And then whenever we think about the environment and everything else, how that can affect a particular phenotype. Yikes, a lot of possibilities, right? Biology, very complex. So the learning outcomes of this week are to be able to differentiate between genotype and phenotype, clearly explaining the relationship between genes and proteins. Explain how most cells in an organism can have the same genetic content, but they can have different functions. They can have different phenotypes. Okay. How is that possible? I still get like, this is a point where I think maybe we don't spend enough time on this, but how can all the cells in your body, they have the same DNA. why is a skin cell in your finger not the same as a cell in your eye? Yes. Okay. Does everyone agree? It's the gene expressions that make them different. Okay. I'm not seeing a lot of opposition there, but yeah, so I think that's it's at the core of things. It's at the core of things. So yeah, you can have, the cells are different. They express different genes. They have different gene expression programs, and that ultimately results in different cell types, different, remember stem cells, right? Stem cells can make all cell types. They can differentiate into all cell types. And so you can imagine the gene expressions in the stem cell, it's all the kind of the same. It's just replicating, make another stem cell, replicate, make another stem cell. But then as the cell gets along and further away from its parent cell, it starts to enter into a different environment, right? And things are different here. I don't recognize it. And so the gene expression patterns start to change. The cell starts to do something different, starts to become a little bit different from the parent cells. And ultimately, as it goes further and further along, it really gets committed to a certain developmental pattern. It can be committed to becoming like a skin cell or an eye cell. So anyways, those expression patterns become different depending on what the cell type is. Okay, so discuss how various factors influence a relationship between genotype and phenotype. It's really interesting how you can take plants. We do it in the lab all the time. We take plants, mother plant, beautiful plant, make a cutting. We cut a branch off, and we cut 20 branches off, and we make what are called clones. They're identical. They all came from the same mother plant. And given a little bit of time, they all look different. They have identical genetics. And then they look different. Explain how two or more genes might interact to determine a trait, so gene interaction, how the environment might impact the phenotype, and how genes and the environment can interact to produce it. Okay, so genes and the environment. Explain how continuous traits are the result of many different combinations. Polygenic, gene combinations. So explain how continuous traits are the result of many different gene combinations. So they're polygenic, many different genes that can contribute to a varying amount of a phenotype. So those are your learning objectives. We're hoping by the end of this class, you'll have a much better grasp than you do already based on watching those videos. Everybody did their pre-class quiz? Good, do them, get the points. Okay, so here's your first clicker. Can two blue-eyed parents have one or more children with brown eyes? Yes or no? Some of you are clicking C or D. I'm going to deduct activity points if you do that. Okay, 5, 4, 3, 2, 1. 78% of the class says, yeah, blue-eyed parents, can two blue-eyed parents have more than one or more children with brown eyes? And they say, yes, this is possible. Blue-eyed parents 78% of the class. How do you guys know that? How do you know? Yeah. Okay. I love that. Yep. Very good answer. But 78% of the class are thinking just like him? I don't think so, because that was a very complex answer, and you're right on, man. Yeah. I love that. I was of the same opinion. Apparently, we're wrong. Apparently, we're wrong. I thought the same as you. No. Well, I don't know, 99%. Apparently, it is possible. I'm just wondering how you guys know all that, because I'm a geneticist. I'm not an eye. I don't focus my studies on eyes and stuff like that. But I grew up on this understanding. The blue-eyed parents, they don't have brown. If one of my friends had two blue-eyed parents and he had brown eyes, I'd be like, sorry to tell you, pal, but yeah, okay, I won't say anything further. But apparently it's possible. Yeah, I'm just surprised how so many of you know that. We'll get into the reason why, but eye color is a lot more complicated than we think. Me and you, we think that yeah, they're just two recessive blue eyes and blue eyes are recessive eye colors, and it's as simple as that. And two people with recessives, they have a kid, and it's going to be blue-eyed. But no, it's more complicated than that. Much more similar to what you were saying. I don't want to repeat it because I'm going to give away a lot of stuff, but we're going to get into it. Okay, so consider your eye. What goes into making an eye color, right? Don't memorize this. There's no exam. There is no exam, but there's no test question. You don't need to memorize the anatomy of an eye. Just go with it. Okay, so you got these different cell layers. This is like a cross section here, and it's showing these different layers of the eye. And this is I guess this is from the top. And what would this be? This is another section, but you can see how there's a lot of different cell types. There's different tissue types that make up an eye. So don't memorize this. This is just to orient you what the heck was in those pictures. Anterior border layer. That's this right here. And posterior pigment epithelium. That's back here. And so there's actually these two different layers that contribute to eye color. All right. And so the anterior is like the closest to your lens, and then your posterior is like that's deeper inside your eye. And those two layers contribute to your eye colors, not just the one layer. Okay, so where is there variation in eye color? Go ahead, talk to your neighbor for a minute. Where do you think the variation comes from? That anterior, you can even look at your neighbor's eyes and see. Is it that anterior, the one in the front, or is it the one in the back, that posterior pigmented epithelium? Just take a few minutes, discuss with the neighbor. If you're sitting alone and you have an open space, tell somebody to come and join you. Okay, so we have the anterior surface and the posterior surface. Okay, the anterior, in this area, there are cells called melanocytes. Have to remember that. The amount and quality of pigment in melanocytes varies between people. And the blue circle at the bottom of the image, this layer is always pigmented, except when people have albinism or albinoism. Albinism. There's little variation amongst people. So, all right, so in the figure above, which layer, top or bottom, do you think plays the greatest role in human eye color variation? Yeah. Yeah. Okay, 5, 4, 3, 2, 1. Okay. 69% say top layer. Okay. And 30% say it's the bottom one. And so I kind of I did give it away for those of you. So 69% may or may not have been listening to me closely, because I kind of did say that was that layer. It was the top layer that is most variable among people. What if I would have changed the question and I would have said, which of these layers contributes most to eye color? How is that question different? This is a really important distinction. Okay, I'll repeat. So here I was asking about which of these layers plays the greatest role in eye color variation among humans, among people. And my second question was, which one of those layers contributes most to eye color? What word did I leave out of that one? Variation. What's the difference? So got a lot of different eye colors. If we, could scan all your eyes and then put them all together in an image, you'd see a lot of eye color variation. And that would be generally what's going on in this layer. Different pigments being synthesized, different organization of the same pigments, differences in the cell structure, because it's not just pigmentation, it's also the way the cells are shaped. Okay, that's going to cause a lot of variation, but unless there's albinism, somebody has albinism, everyone in the class will actually have a pigmented layer in behind. It's not varying. It's contributing a lot to the eye color. Those black lines you see in the eyes, but it doesn't vary. There's no different pigments being synthesized there. Okay, so what do you think? Do you think that one gene contributes to all of these different layers? Sorry, I'm trying to minimize some stuff here. Do you think that one gene contributes to all of these different layers? No. Somebody said it. No, I don't think so. Does everyone agree? So we got different layers. Imagine you're a stem cell and you're going off into the developing, being a replic. Geez, I don't know. I'm having a hard time talking today. You're developing. You've been replicated. You're being pushed farther and farther away from the stem cell. You're being pushed into an area that's going to develop into a certain tissue type. The cells that end up here, down here, going to be different than the cells here. Different in shape, different in function, different in pigmentation, different in all kinds of respects, right? So you wouldn't think that it'd be one gene that's responsible for the different layers or even the pigmentation in the or the color, the phenotype that comes from the different layers. All right, good. We got that. We got the whole polygenic thing being emphasized. That's great. Now, let's talk about categorizing things. So on your own this time, on your own, let's sort some of these colors. Okay, so got some Google slides. You're going to look at images. You're going to look at images of eyes, not these colored things. You're going to look at images of eyes, and you're going to sort them using into whatever groups you think they belong. So I want you to sort them based on eye color into discrete groups. Okay, into discrete groups. So we're going to do that first. I'm going to open up that activity. Just give me a sec here. Yeah. That's today, yeah. Okay, so if you look on eClass now, there should be week 3, January 24th, or it's just WK space 3 if you want to use your Control F to find that. But anyways, it's in the week 3 section. It's of course I just lost it. Week 3 activities right here. Yeah, that's it. Oh, is that the this class? Okay, whenever you click on that, let me click on that. What does that do? Oh, okay. Are you guys able to pull up the activity slides with that one? Okay. It's not allowing me to log in. So.
Hello, Professor. I just wanted to state that Zoom doesn't have the video.
Is anybody getting a heavy traffic message? Yeah, just give it a few minutes. Okay, so I don't know, is it maybe maybe the link is not working just yet for some of you. You can always come back to it later. But many of you have gotten in, and if you've done it, you sorted the eyes, just drag and drop. You sorted them into groups, okay, on your own. I'm sure there was no discussion about how you were sorting them. Now what I want you to do is I want you to get in groups of 2 to 4, and I want you to compare your answers. How did you sort them compared to how your neighbor sorted?
Professor, excuse me.
Okay,
Excuse me, Professor.
clicker. Did all the members of your group have the same number of categories? Nah. There's only 2 options. It's yes or no. If you put a C or a D or an E, I'm deducting points. Oh, I see an E in there. It disappeared. Okay. All right. 5, 4, 3, 2, 1. Thank you for participating. 58% of the class, the majority of the class said, no, we didn't come up with the same number of categories. I'm impressed. 42% did say that they had the same number of categories. So that's not what I would have expected, but yeah. Now, if you look carefully at your results compared to your neighbors, did you always put the same eyes in the same group? Okay, 5, 4, 3, 2, 1. 58%. You know, I think you guys are just pushing the same buttons. You guys aren't even, you're just eating a sandwich and pushing. Guys. So you said, first of all, 58%, like, we don't even have the same number of groups. And now 58% says, yeah, we put, or no, they say, no, it was only 43%. I got it backwards. 43% are saying, yes, we put the same eyes in the same groups. Wow, that's impressive. You guys are really tuned into one another. But chances are it wouldn't happen like that way. First of all, you wouldn't come out with the same number of groups. It's kind of subjective, right? And then would you actually put, the eyes are so close, and you would actually put them into the same groups? Not likely. You would need to really well have a very clear, strong definition of what belongs in one group or another in order to be able to do that, in order to place the eyes into the same group as another person. That's an important point, if you think about it. You're doing research, and you call one group the blue-eyed group, and the other group the brown-eyed group, and you publish in your study like how many people from, I don't know, whatever, whatever the study was about, where blue eyes and maybe they had CVD. How many people with blue eyes had CVD in this group? How many people with brown eyes had CVD and end up in a group? And you would have to very, very clearly define what the difference is between the groups. So when you have ambiguity, the eyes are not quite blue, they're not quite brown, it's somewhere in between what group it belongs in order for somebody to be able to reproduce that study, in order for people to come to the same conclusions that you did based on your study. So that's very, very important. We make categories up kind of subjectively sometimes, even though there is no clear separation between groups of things. Okay, so quantitative versus qualitative variation. What do these terms mean? I think you've all heard this before. Let's go over it again. Okay, so this is a pretty good slide to think about it. So you got quantitative on the right, and you got qualitative on the left. Qualitative here has discrete, separate categories of phenotypes. This is like blood type, right? You have the A antigen. It's clearly distinct from the B antigen. And then you can have A, B together, or you can have none. And there's a discrete, they're discrete. So there's a clear separation between them. So they easily fall into these observably distinct categories, let's say. All right, but quantitative variation is now you have like a gradient, a continuous gradient of possible phenotypes. So it's a quantitative trait is the one where you have an observable phenotype that varies over a range, a continuous distribution. You can imagine just a simple example is like tall and short. That guy's tall. No, he's short. It's a continuous thing, you know, 5'1", 5'2", 5'3", 5'4", 6'1", 6'2". It's a continuous gradient, right? Height is a continuous phenotype, and so there can't be. You could subjectively put people into tall and short classes. You could do that, but it's actually a quantitative trait. And this figure actually sums it up pretty nice. Like if you look, they're just looking at 3 genes here, the A gene, B gene, C gene, okay? Possible offspring from these 2 parents here, these heterozygous parents. And they're showing you a gradient of the phenotype. I guess the phenotype is color, whatever that is. But you can see here in triple homozygous, I want to say recessive, but I'm not going to. Allele type, you can see a very light color. And then if you have one dominant or one type of allele for the one gene, you can get a darker color. 2 alleles can give you a darker, and so on and so on and so on, until you have all of these particular types of alleles, and you get a really dark color. Why am I being so hesitant to say dominant or recessive? Yeah. Okay, dominant or recessive doesn't mean that it's inferior or, yeah, not really understanding. Yeah. Yes. Dominant can be picked over recessive, but recessive not over dominant. Okay, not 100% getting it still. Yes.
Like excessive or applies to qualitative variables. And in that case, it's one gene relation that you really have clause to apply. But here in quantitative variations, multiple genes are involved. So it may not be specific like domination by one gene or the other. So maybe Gillian laws do not apply. So it's hard to really
I want to give you a microphone so I don't need to. This is excellent answer. Excellent. And I'd have a hard time reproducing it exactly the way that you said it. That's very good. Yeah. So dominant or recessive, it's a relative thing, right? And it's more easily, it's a relative thing. The phenotype is what tells you whether something's dominant or recessive, right? Is that true? It's the phenotype, right? I don't really have a dominant allele or recessive allele. It's more so about what those alleles cause on the phenotype. So the phenotype, particular allele. So if one allele is present and it tends to show a particular phenotype that can mask another phenotype, we call that allele dominant relative to the other allele. It's a relative thing. And it's a relative thing, and particularly you can see it more so in qualitative traits rather than quantitative traits, because quantitative traits, there's a gradient of possible phenotypes, right? Like you can imagine, here we have one uppercase A, and everything else is lowercase, and we get this color. What if we had one uppercase B? And everything else was lowercase? Would we get the same phenotype? Maybe according to this. If we had one uppercase C and everything else was lowercase, would we get the same phenotype? Okay, so now dominant versus recessive, it's a relative thing. It's relative based on the phenotype that you're looking at. And it applies more so to qualitative traits, not to quantitative traits. Okay? Sorta. It's more clearly seen in a qualitative trait. So, for a quantitative trait, why is there a range of eye colors beyond just brown or blue? Why do you think that is? Why is there a range of eye colors? Yes. Very good. That was so very complex answer, but that's the truth, is that this is a polygenic trait. There are many genes contributing to eye color. Look at the variation you see. There's no clear blue and brown. There's like a gradient, right? A continuous distribution of phenotypes. So generally speaking, most phenotypes are like this, okay? Most phenotypes that are quantitative are polygenic. The reason why they're quantitative is because there are many genes that are contributing to that trait, many different alleles, and having one particular set of allele at one gene is going to affect how the other gene contributes towards that particular phenotype. Imagine if you have 10 genes. Well, now that's getting complicated. We just looked at 3 genes when we looked at this color. Look at all the differences you can get just with 3 genes. Now imagine you have 10,000 genes. That's a lot. And that's why genes interact, the products interact, the phenotypes are complex because many, many genes contribute to them. This is the norm. Okay, that's why it's very difficult and why there's such struggle to, after you realize that, to go with the simplistic idea of Mendelian genetics and how it was traditionally taught and how it's taught in textbooks. Okay, so many genes contribute to eye color. Is this quantitative or continued? Is this quantitative or qualitative? It's quantitative. There's continuous variation. Do you think one gene contributes to all these layers? You already told me no. Guess what? Polygenic. Polygenic means the number of genes contributing to a phenotype. That's the norm. Okay. So many genes influence the composition of each of these layers. And the layers themselves interact to form a particular color of particular eye phenotype. Okay. Okay, so they not only influence the pigments that are present in these particular layers, but also the way light's reflected, the way light's absorbed, and so on, and that further influences the phenotype. Okay, so why did we think that it could be as simple as this? You know, two brown-eyed parents, and they're going to have definitely brown-eyed children, and maybe a recessive come together, and they make a blue-eyed or green-eyed child. Whenever it's actually very, very complicated. You can see even this person, half of their eye is brown, and the other half is blue. It's really much more complicated, right? So what we've been taught is it's a dramatic oversimplification of things. All right, so we're going to look at fruit flies now. Yay. Fruit flies. Actually, they're pretty cool. Go ahead, put your answer in the clicker. Okay, 5, 4, 3, 2, 1. Okay, good. I was wondering if you guys can see it very well because, okay, the lights are shining on there. 74% of the class say no. Those eye colors are, those are different eye colors. Okay. Yeah, I agree. I see two different eye colors there, right? Okay. Now, how many genes play a role in this orangey kind of eye color? That's what we're going to look at. It's an eye color phenotype. Relative to the wild type, this is like a brownish. This one here is an orangey color. This one's a brownish color. Wild type, you know what wild type means, right? What does wild type mean? Whenever you just in research, you always see wild type this, wild type that. Yes. Okay, yeah. Yes. Normal and being in a population, that's kind of your standard. If you were to breed for generations and generations, this is what the phenotype that you would encounter, this wild type phenotype, right? And okay, so the eyes after generation look like this, and then we have some rare mutants like this one here, vermilion gene mutant that gives us this particular orangey color. So in this population, you'll find a very low proportion of flies that have this orangey color due to a mutation in this particular gene called vermilion. Okay. Now, are these two fruit fly eyes the same color? Five, 4, 3, 2, 1. 78% of the class say, yep, Those two, they look like they're the same color. Okay, so what I just showed you was the vermilion mutant and the cinnabar mutant. Okay, these are two different genes that are mutated. And so both are giving you this, this orangey color compared to the wild type. But they're different mutants, right? They're different genes that are mutated. So you got your wild type here, this brownish color, and then you get this orangey color here. So how many genes play a role in the orangey eye color phenotype? Can we answer that question based on the information that we have? I see some head shaking. Thank you. No. Well, do we know that there's at least two genes? Sorry, this is kind of a trick question. Do we know there's at least two genes that cause the orangey color? Okay. All right. Just hold on a sec here. Just hold on a sec. So I want you to think about the way genetics works and what you're seeing here. Normally you have a brown color and you get a mutations and you get an orange color. Do those genes contribute to the brown color or to the orange one? Oh, now let's change our answer here because I guess did you guys get it? Did you hear it? So what color do the vermilion and what color are they contributing to? The brown or the orange? Brown. The brown color. When the gene's functional, vermilion gene's functional, this is a weird thing geneticists do. They give phenotypes, they give the genes names based on the phenotype, but the vermilion gene and the cinnabar gene, they're not involved in making orange. You see orange when they're not working. They're involved in making the brown color. This is the kind of stuff I think it's kind of interesting. I like pathways and stuff like this. So, but because that's the way a lot of genetics is. All kinds of genes are contributing to building this final product, which is going to be partly responsible for a phenotype. So there's actually two different pathways here. There's this omicron pathway, which makes the brown pigments. And then there's this drosopterin pathway, which makes the red pigments. Okay, right. And so what we're saying here is, okay, imagine the wild type, it has both brown and red pigments or orange pigments. And whenever you mutate the vermilion or the cinnabar, you're actually losing the brown pigments and you're getting the orange color. You're having this remaining pathway still active. I hope you all see that. Okay, right. So to understand eye color, you have to expand your data set and you have to understand multiple genes, multiple alleles in two different pigmentation pathways. So there's at least two genes that are involved in making this brown pigment, not the orange one, the brown one. At least two, because we know if we mutate either one of them, we lose the brown pigments. Okay, to make things more complicated, it's like a pathway of enzymes. What do enzymes do? Enzymes, they convert one metabolite to another. So they make up a pathway. They make up a pathway, right? And so this enzyme is going to take like one substrate. It's going to take one chemical, convert it into a new chemical. And a second enzyme is going to take the product of that enzyme. It's going to convert it into another molecule and so on and so forth until you get the final pigment being formed. It's the same, similar for pretty much any metabolic pathway. Now, so if that's not complicated enough, you can imagine these pigmentation pathways, the brown pigment itself could have 20 genes. We work with glyceolins, I mentioned it earlier. There is at least 25 genes, so 25 enzymes that take the sugar from primary metabolism and convert it into this anti-cancer molecule, the glyceolins. At least 30 of them. So you can imagine a stretch of these guys, 30 long. And so you can imagine any different allele in any one of those 30 genes can impact the production of the final product, can impact the final phenotype, how much drug is being produced for the planet's defense, how much defense it's actually being able to put up against pathogens. And to make things more complicated, there can be feedback, feed forward, all kinds of mechanisms. So if this particular allele of this enzyme is generating too much product and the subsequent enzyme in the pathway can't deal with that much product, you'll have an excess of this particular molecule. And that particular intermediate, it can go back and it can inhibit the function of another enzyme, further affecting the pathway. This is just to emphasize how there can be multiple layers of how the different alleles of genes, of enzymes, isozymes, how they interact to make a final phenotype. Okay, so in this example here, we close down the Omicron pathway. Omicron pathway. By each of these particular mutations, different genes shut down the same pathway, and then we ended up shut down the brown pathway, and we ended up with red pigments. All right. Yeah. If you had to sort these into two groups, how would you sort them? There are many options. This could be one option. A, I'm going to sort them like this. I'm going to put this in one group and these guys in another group. Or B, I'm going to put these two in one group. I'm going to put these two in another group. Okay. Let's look at the genotypes and consider this. We're just looking at this one gene, okay? And let's look at the different groups. So you have this one group here and you have another group over here. And now look at what's going on here. What do you notice about this? Look at this BB genotype. Well, I got a brown eye phenotype here and I got BB genotype. I got an orangey, because I put them in the orange category here, and I got a BB genotype. If you were to sort them like this, would you say that having this recessive B allele has any contribution to the color of the eyes? Does it determine the eye color? No, because you actually put them in two different groups. Same genotype, and I stuck them in two different groups. So it doesn't determine what group it goes in, right? It doesn't determine the eye color. But if I did my groups like this, oh yeah, I see it. I said, these BB things, they're the same color. They fall in the same category. And that's different from this whenever there is a different uppercase B allele present. So if you did that, you would probably be more convinced that B is responsible for the eye color. The importance of all this is that depending on how you make the groups would influence whether you come to the conclusion that a particular gene or gene's allele is involved in determining the phenotype difference between the variation between those groups, right? There wouldn't be any variation if we had differences in that gene and they all ended up in the same group. That gene's not responsible for the variation. So what I'm saying is it's really important how we draw the groups. Okay, how you consider the group CVD, for example. Okay, so to make things even more complicated, the differences in phenotype, you can have identical. Remember I talked about the clones. I took my plants and I made clones genetically identical, but they perform differently. Differences in genotypes can be observed at different times. Maybe at day 0, these guys, they look very, very different, but then later on, they kind of just look the same. At 7 days, they look the same. So the point is that anything that affects the expression of this trait over time can also impact that final phenotype. Okay, so it could be timing. Timing is really important. Whenever we are trying to stimulate glyceolin production, we treat with a pathogen, we treat the soybean roots with a pathogen, and bam, the production turns on. It's only transient. We haven't figured out why it's only transient. Is there feedback? Is there enzymes that degrade it? We don't know, but it's transient. Goes up, comes back down again. We have to look at exactly the time point, 24 hours, whenever it's peaking. Otherwise, we would miss it. Yeah. Yeah, sure, yeah. Yeah, like the eye color, how it kind of takes time to come to fruition. Yeah, very, very much so. Yeah, so timing and you can imagine how environment is like impacting the expression of the genes over that time. And so this is a very simplified network of genes that are contributing to a phenotype. Look at how many of them are. So in other words, if you were to pull on, if you were to mutate, if you were to alter the expression of any one of these genes, the whole network becomes perturbed. And the phenotype will change accordingly. That's real life. That's how biological systems work. And so genes are impacting the function of each other. They're alleles. One allele in one gene, it can impact the different alleles in a different gene. It'll impact the other gene differently depending on what alleles are present. So every time you stack different alleles of genes together in a function, they're always going to interact differently with one another depending on what allele set the other gene has. To make things more complicated, look at this. This is just environment. Genetically identical plants, one grown on acidic soil, one grown on neutral pH, and look at the difference in the flower color. Identical genes. This is the environment that's causing this difference. Okay? The environment causing difference in the expression of the pigmentation, causing the difference in the acidity of the vacuole that results in a different color. Okay, anyways, that's it for today. You have a deep question that comes out. Remember, you have Thursday's class to work on it. Okay? Thank you, guys. Hey. Yeah.
It's not working.
You're what?
My partner, it's not working.
Ooh.
Wh-